Hello everyone, welcome to Get My Class. So let's learn this topic that is DNA fingerprinting, and this has been taken from your chapter that is molecular base of inheritance. So let us understand what is DNA fingerprinting. DNA fingerprinting was a methodology which was actually developed by a person called as Alex Jeffrey. So what he did, he used satellite DNA. Satellite DNA consists of the repetitive sequence, and during centrifugation, these repetitive sequence get separated from the rest of the DNA. That's why these are called as satellite DNA. So he used satellite DNA as probe to show high polymorphism in the sequences called as variable number of tandem repeats. That means among repetitive sequence, again there was variation in the sequence, okay, sequence of the repetitive DNA, okay, or repetitive part of the DNA. Okay, so these variable number of tandem repeats, these the size can be between zero point 0.1 to 20 kilobases. Okay. Now the polymorphism, when we say polymorphism, that means the nucleotide sequence, there is slight variation in the repeats itself. Okay. So polymorphism in the DNA sequence is the basis for genetic mapping, because this polymorphism, that is the difference in the sequence of the repetitive DNA, that will make me and you separate. Though we are human beings, but still, though we are 99.9% the genome is same, but still that 0.1% there is difference. So in that 0.1%, this polymorphism is one of the factors which separates individual person. Okay, so it is necessary for genetic mapping as well as DNA fingerprinting. And it also helps to know the difference in the DNA sequence of repetitive DNA. Though it is repetitive DNA, but still there is slight difference in that, and that's why we call it as variable number of tandem repeats. The polymorphism is, it happens, of course, there is variation at genetic level. That is what is polymorphism. It arises due to what? Due to mutation. Suppose you have a particular type of sequence which is repeated everywhere, we call it as a repetitive sequence. But even in that repeated sequence, there can be slight changes in the nucleotide sequence. Okay, maybe due to mutation. Okay, now allelic sequence variation described as DNA polymorphism. If more than one variant or one allele at a locus with frequency greater than 0.01 occurs in human population. Okay, so if in human population, if the allelic frequency is greater than 0.01, then it is considered as DNA polymorphism. Now, DNA polymorphism, it is an inheritable mutation because the mutation is happening in the DNA, and of course that genetic information will be inherited by the next generation. Okay, so it is inherited mutation and it is observed in population at high frequency. And this is how the, you can say, DNA fingerprinting is done. So specific chromosome, they will have specific repeats, that is variable number of tandem repeats they will have. So here they have taken the chromosome number 7, chromosome number 2 and chromosome number 16 from a particular individual, that is individual A. And the same numbers of chromosome they have taken here, chromosome number 7, 2 and 16 from another individual. Okay. Now this is the, suppose they want to find out like who is the criminal here, whose sample was found in the crime scene, and they have the details of the sample which was collected in the crime scene, suppose. And this is the bands which have been formed based on the number of short tandem repeats, or variable, you can say variable number of tandem repeats. Okay, of person A here and of person B here. And this is the tandem repeat bands of the person whose sample was collected from the crime scene. So now they will match up these suspects, that is A suspect and B suspect, with the sample, okay, test sample. Now you can see that the B suspect's DNA fingerprinting is matching exactly the C one, okay, that is the test one. That means B person is the culprit, whereas A person, the tandem repeats are not matching. Okay, so this is what is called as DNA fingerprinting. Okay, based on the number of short tandem repeats, or based on the variable number of tandem nucleus. Okay. Now, now there is, I told you that there is something called a satellite DNA. So what is satellite DNA? As I told you, it is nothing but the repetitive DNA itself, which gets separated during centrifugation by density gradient. So the rest of the bulk genome will form one particular peak, whereas you have short, minor peaks which are formed by these satellite DNA, okay, which gets separated from the rest of the genomic DNA. So that is why they're called as satellite DNA. And what are they made up of? Of course, they are also repetitive DNA. Okay, and VNTRs, that is variable number of tandem repeats, are type of, or you can say part of satellite DNA. Then satellite DNA can be classified based on the composition, like what sequences they have, as well as based on the length, like how lengthy they have or what is the length, the number of repetitive units. So how many units have been repeated again and again. Now, for what is this DNA fingerprinting used? It is used like, the samples of the DNA where every tissue, that is the DNA samples from every tissue like blood, hair follicles, skin, bone, saliva, sperm, etc., can be utilized for DNA fingerprinting. Now, it will be helpful in solving disputes related to property and biological relationship. Okay, if we have a person who says that, that is my child, pointing out to somebody, so we can do DNA fingerprinting of the child and the father, and we can say, whether he is the actual father or he's just, trying to take away the child. That is paternity testing can be done. Then used to compare the DNA sequence of any two individuals. So any two individuals can be just, if you want to see the comparison, how different they are, that DNA fingerprinting will be helpful. Then establishing a link between biological evidences and suspect in the criminal investigation. So these are all the application of DNA fingerprinting. So finally, we are going to talk about the protocol which is followed during DNA fingerprinting. The first step, of course, is the extraction of the cells by high-speed centrifugation. So you have to extraction of the DNA from the cells. So you have to extract the DNA from the cell. Okay, then amplification, DNA amplification by PCR. Okay, so you will amplify that particular um, you know segment of the DNA, or that the DNA what you extracted. Further, DNA fragmentation by restriction endonucleases. So what we do, we fragment, or we can say we will digest the genomic DNA into small pieces. Okay, so that which will allow us to easily separate those pieces in the form of bands using gel electrophoresis. And further, double-stranded DNA split intos DNA, that is single stranded DNA. That means double-stranded will get separated into single-stranded DNA. And now what will happen? There is southern blotting. That means whatever DNA is present in the gel of the gel electrophoresis, it will be transferred onto the nylon sheet or nitrocellulose membrane. Okay, so the DNA from gel electrophoresis will be, or from the gel will be transferred onto the sheet of nitrocellulose membrane or nylon membrane. This is called as southern blotting. What is this called as southern blotting? That is transfer of separated DNA onto nylon of nitrocellulose membrane. Then you have hybridization, okay, or probing, you can say, where nylon membranes are immersed with probes or markers. Okay, so probes or markers, these are all radio labeled markers which will go and bind to specific bands which are present on these nitrocellulose membrane or nylon membrane. Okay, so that they will hybridize with, the probes will hybridize with specific sequence there on the DNA fragments. Then we will look for, like we will go for autoradiography, because the probes which we have used, they are radioactive probes. Radioactive probes. And they will show us probes, right? Yeah, probes. They will show us the position where our specific sequences are present. Okay. Then further, the nylon membranes are pressed on the X-ray film so that these radioactive probes will emit radiation and there will be markings which will form on the X-ray films. Then at probe site, dark bands appear. Okay. Then probes or markers. Here, the radioactive synthetic DNA and is complementary to VNTR. So they will find, which is complementary to VNTR. They will find out the exact sequence of VNTR and how many numbers of these repeats are present. Okay. So this is the entire protocol of DNA fingerprinting. I hope you understood the explanation. Thank you.